A very good morning to everyone. I, Meenu Agarwal, Assistant Professor in Department of Biotechnology, welcome you all for joining us on the second day of International Conference on BioTrendz, Advancement and Challenges in Science and Technology conducted by Department of Biotechnology, NIET. Now, I'd like to introduce our first speaker for the day, Professor Pawan Dhar. Professor Pawan Dhar is currently the Dean, School of Biotechnology, Jawaharlal Nehru University, New Delhi. Synthetic Biology Group, School of Biotechnology, JNU, New Delhi. Professor Dhar received his PhD degree in 1993 from BHU in the area of human genetics. Professor Dhar has launched and incubated the first biotechnology company in JNU to deliver therapeutic molecules from a novel drug discovery platform. Prior to this, he held senior scientific positions at Shiv Nada University, Riken Genomics Science Center, Japan, Bioinformatics Institute, Singapore, Keio University and Kyoto University in Japan, and Manipal University. In the past, Professor Dhar has headed Riken Japan and TCS Hyderabad program on metagenomics, established research lab in India, Singapore and Japan, and has a total of more than 80 national and international publications in genetics, biotechnology, bioinformatics, system and synthetic biology to his credit. He has industry collaborations with Intel, Bangalore, and Tata Consultancy Services, Hyderabad. Professor Dhar serves on the board of referees for European Science Foundation, Executive Council member, Chair Intellectual Property Management, member Biosafety Committee, academic committees of CCMB, NII, and Indian Naval Academy. DBT, Task Forces of Marine Biotechnology and Genomic Engineering. He is founding editor-in-chief, Systems and Synthetic Biology Journal, Springer. We are pleased to have you here, sir. Please enlighten us with your thoughts.
Thank you very much, ma'am, for your kind introduction, and a very good morning to all the participants who have joined in this international conference. So today I'll be talking on systems and synthetic biology, and this is very recently come up in the community. Hardly, the last 1.5 decades, we have been seeing some advancement in these areas. So my intention is to walk through the fundamental concepts, and we'll try to see what was the reason for systems and synthetic biology fields to come into existence, and what kind of advancements have been there for the past few years, and also touch upon our work. So reductionism has been the key scientific approach in doing science. Reduction means that you have an organism and you want to know how it works, how it functions, and then you want to search for an answer within the organism. So you have to go deeper into the organism in order to understand how it functions. So this approach of going from the large scale to a smaller scale is called reductionism. And when you look for an answer, then you come across these organs, these systems that are stitched together to create the whole organism. And now, when you have a reasonably good view at this level, obviously you want to know what's there deeper at the deeper level, because these do not give you enough answers. So you're looking for answers. So you go to one level down, and then you come across the cell, and this cell, if it is stained in a normal way, then it looks an empty structure with something in the middle, and then you investigate further based on your technology development, and then you realize, and the picture on the right corner, you realize that the cell that seemed to be very innocent is actually full of details. And now your technological aid and staining techniques, they both help you to investigate further. So when you go deeper, people came across this structure called chromosome, and they found that DNA needs to wind itself 6,000 or 7,000 to 10,000 times to make a chromosome and unwind itself to make chromatin. So this was is a statistically precise activity, a mega activity at a micro level. So earlier, when DNA was identified as a big molecule and kind of a superstar in the community back in the 50s, people thought of DNA as more of a director of nuclear affairs, that it is the molecule, it controls everything. But over time, RNA community and the protein community caught up the attention. And so over time, what has happened is that DNA has become more of a database of information rather than the regulatory molecules. So now when you go a little deeper from DNA, you come across chemical reactions. Now these are chemical compounds talking to each other. So now you leave typically the biology and you enter chemistry. And if you go deeper, you come across atom. And when you go a little more deeper, then you see that an atom is 99.99% space. And you cannot go deeper because how do you view the space with a microscope or with something else? Therefore, when you're trying to understand biology and you're going to that level, you understand that as you go deeper and deeper, you're losing resolution or the control over information. And so when these atoms talk to each other, chemistry is born. And when chemistry comes into biology, and then you see biochemistry and other forms of biology, fields of biology. And so overall, what we have just seen is this process called reductionism. And this reductionism is giving rise to components, and we call them as genes and proteins and RNA, carbohydrates, lipids, and so on. And this particular method of generating data from the cell worked pretty well, and as a result of which we have a large body of data. Now, in the mid-90s, there was a big problem, what to do with this data, because it's a lot of data. So then the era of capturing the data in the form of databases started, and the practice of modeling the data. Modeling means that you're creating a representation of the data in the computer. And so even a database is a model in that respect. So after the modeling, then there was another thought. Early 2000, 2001, this thought came up because people said that now we know how to reduce the information, how to split open the system and get the components. That is one way of doing things. The other way is to capture these components in the computer and do experiments through computers. So that's another way. Could there be another way, the last one, of designing the systems that did not exist in nature, or they exist in nature? You're constructing the systems. So the first approach is commonly called as molecular biology or biochemistry. And the second approach is more often recognized as systems biology, or at component level, you can say bioinformatics. And the third approach is what we call the synthetic biology. So just want to get the definition right before we delve deeper into these fields. One is about reducing the information. The second is about capturing the information. And third is about creating a new information, right? So you have three different fields. So the integration of information has led to creating virtual red blood cells. This was the project that I joined in Japan back in early 2000. They published, in fact, late 90s, they published a paper in bioinformatics where the group led by Masaru Tomita, for the first time established that one can transfer biological information in the form of pathways to computer, make predictions, and they can be experimentally validated. So likewise, people are now looking at the possibility of making virtual skin that can model the growth of the hair, the color of the skin, and the aging process together can be captured in the same model. And this is a big project in the US and Europe, the Virtual Brain Project. which is taken off very well, and a lot of information has been derived from here. New technologies have come from this project. I'm not trying to oversell the modeling because it can lead to incomplete understanding, which means that the real object is the real gene, protein, system, cell, tissue, organism, whatever you call it. But when you capture it in the form of a computational equivalence, in the form of a database or in the form of a pathway model, then it becomes a representation of the system. It's not the system itself, but it's a representation of the system. And as you can clearly see, the representation of the system can be measured nicely. Say the shadow can be measured quite well by length and breadth. But what has happened is you lose a dimension, right? So information loss is essentially an activity that you come across while modeling the system. So you don't model each and everything in the system. You model only the key components and hope that these key components will represent most of the system behavior. Now that's the art of modeling. Sometimes it can also lead to incorrect understanding. So what you model, what you got in the computer may not match what is there in the real system. So you got to revise the model, you got to revise the values and parameters and so on. So this is the thought I proposed, quite some time back. Is it time to construct a periodic table in biology? And if yes, what would be the components of this periodic table? Because if you have a periodic table representation, accurate representation, engineering of biology gets easy because you start at the molecular level and you can assemble the components and predict what is going to be constructed. So it has a payoff, but then there's a huge information gap above and below the fold level. So this will remain for quite some time. And so the question is, can biologists think like engineers? Why are we thinking like that? Because there are similarities between biology and engineering, as you can see on the screen. However, there are also dissimilarities, especially the fact that if you design engineering systems, you're talking of components that stay there. They don't move all the time. Even if they move, they're glued to something. They're latched to something. So there is a certain firmness to their address or location. While as in biology, you have RNA, protein components that are moving all the time. They don't stay at one place. So how do you hardwire these molecules that are moving all the time from one compartment of the cell to another compartment? They are moving. So you've got to have a different engineering approach of stitching their properties together. Engineering systems are predominantly digital, like the ones that we are using today, the computers. But biological systems are predominantly analog, and we'll see a graph on that. And there is a unidirectional flow of information in digital systems, while as in biology there is a reversible flow. So, for example, as you can see, the enzyme substrate complex. The enzyme substrate complex itself is an indication that some of the information from the enzyme substrate complex goes back to the substrate. So there's a reversibility of the information, which is not found in the engineering system. And Mendel's laws are the only ones that we know. If you call them laws, some people like to call them as ratios, then laws. But at the end of the day, they don't help because they are zoom in, zoom out types. So they zoom in from phenotype to genotype and genotype to phenotype. So they don't help you in designing molecular systems. All right. So in this background, when you got to make something, then you have to consider that your systems are noisy and unpredictable. So noise means there's a fluctuation of the gene expressions at every time point, and unpredictability because we do not know what's going to happen if you remove or add a gene. And if you have to make a biomolecular circuit, the information is lost at many steps, at 2 steps at least. Before the information reaches the next point, it has to go vertically downwards, survive certain filters, and if the information survives, then it goes to the next step. All right, so it's not very, very easy to construct a biomolecular circuit as against the engineering circuit. So to design well-behaved system, one needs standards and rules of composition. Now, the standards are the ones that remain fixed. It does not mean that standards are some God-given features. No, standards are the ones that are made by humans. So for example, in this case, the standard that was made in the broad gauge, in this particular example, is 5 feet 6 inch. It could have been 6 feet, it could have been 7 feet. Well, somebody fixed it because of some reasons at that time. And then after this, you have a meter gauge, you have a narrow gauge. That also is possible. So standards are something that you fix them once for all. We do not have these fixed quantities in biology most of the times. And then we need to put the molecules together to construct higher order system. Then we have rules of composition. So we compose the system in a certain format. You cannot put the parts together randomly and hope that a system comes up. You have to be very precise in what order do you stitch these parts together. So you got to know the standards and the rules. If you know both, then you can construct biological systems. And the grand challenge is with the chemical synthesis of cells using rational design approach. Now, IEEE standards that are very common in engineering do not exist in biology. Therefore, overall, it is good to use engineering as a philosophy. Then we don't have any tools. So you got to make those tools. The reason they don't exist is because probably engineers never faced a problem. Now, this is not to say that what they are doing is less complex to biology. It is just to highlight that there's an uncertainty, emergent phenomena, probability, gradients, and the amount of new information that comes up. Emergence means that something happened with the same group of molecules that was not predicted before by any computer. It was just not possible to predict it, but something new happened. This happens in biology all the time. Likewise, when the transcription factor talks to DNA, then it is not an assured interaction. This may or may not happen if the transcription factor is not close enough. Even if it is close enough, there may be some molecules in between due to molecular crowding. So therefore, nothing is assured in biology. Everything is a probability, right? So that is what I'm talking about complexity, and not the number of components, but uncertainty of component interaction. So what's the solution? Practical solution is that we have been good in decomposing the system one part at a time. Now let us compose the system, but this should be a regulated construction, a controlled construction. BioTruth table was proposed back in 2004 when the first event of synthetic biology was organized at MIT. BioTruth table was proposed as a way to go. So for example, if you had to design E. coli, construct 4,000 truth tables and see if we can put the data together in the form of model, or the yeast 6,000 truth tables. But it didn't happen because in reality, we can say logic gates is the way to go. We can make it, sort of an analogy. But in reality, the rocket is never straight. It's not a straight rocket science, not a very deterministic rocket science. There's a lot of probability. So what we end up is making a different version of the reality, that what we hope. So synthetic biology is a rational design and controlled construction of biological components leading to applications. People are also calling it biological engineering. In fact, large, now members of the community call it biological engineering rather than synthetic biology, because obviously it is an engineering approach. Just want to highlight for those who are in India, so we do have this special feature that was started a few years back by the Department of Biotechnology and JNU. And now it has evolved into a bigger scale of involvement. So teams from India, they submit these entries every year to the Department of Biotechnology. And we review those applications, and we select 5 teams typically and give them 10 lakh budget each team. And then this team then goes to MIT and presents the work. And so what you see is the picture right now is at MIT. So many teams from India have gone there. They have won silver medals, gold medal, and done phenomenally well. So if you're interested, I think you should have a look at IGM website and also DBT website. Well, we have a very simple mission. We want to create brand new genes from and brand new proteins from what is called as the dark matter of genome, which does not code for anything, no proteins, no RNA, right? So to provide the proof of the concept, we took intergenic sequences. As you can see, the red colored lines, we took different sequences in different directions, different sizes, overlapping, non-overlapping, and we took all of them, and then we did not know what to take because there is no publication to this. So we took it for the first time. And when we found, so we can, how do you construct a brand new gene from an intergenic sequence? is when you take a non-expressing DNA sequence, an intergenic sequence, and you install these signals upstream and downstream. And therefore, by installing the signals upstream and downstream, you can construct the gene. But how do you do it? Where do you do this? So, because if you look at the genome sequence level, then this genome sequence is it's complex to do all the edits there. So we pulled out the information out of the genome and used a certain vector system. So what you see in the next slide here. So we pulled out the information once we bioinformatically clear that this is a brand new sequence which does not have a protein sequence equivalent in nature. We took an intergenic sequence, amplified it, inserted in the vector, expressed it, and the vector itself provided us all the infrastructure for expression. As you can see here, the pBAT topo expression vector by in vitro gen was used, and there we go. So the best moment was found when we expressed these and we could find the proteins on the gel. In the Western blot, we could find the proteins. And this was for the first time that somebody was making proteins from intergenic sequences. So the phenotype of the transformed cell, we also checked the structure, but we checked the phenotype, and that was a little more interesting. Because of all the proteins that we made, one protein expression was consistently killing the cell. So we said, if that is true, if the killing of the cells is true, then if we switch off the gene expression, can we restore the growth? So then we found that the growth is restored by switching off the gene expression, as you can see in this slide, by switching off the gene expression. So first of all, the cells are getting killed after 2 hours. And when they are killed, you switch off the gene expression and the growth is restored back up again. So thereby proving a linear correlation between the protein expression and the cell growth. The functional study of the lab-made genes, as we call them, is overexpression. If you underexpress them, or if you do not express them, it doesn't matter because nature never recognizes their expression anyway. So then this led to making of an Encyclopedia of Lab on genes. When you profile intergenic sequences, introns, pseudogenes, every sequence that you thought is never making proteins, we took those sequences and we said, let's make proteins from them and see what happens. The next few slides will show you the experimental and computational results. So we found a novel antimicrobial peptide coming from the yeast intergenic sequence. And this antimicrobial peptide was resembling an existing peptide, which is defensin molecule. And in terms of structure, they were strikingly similar, almost 98% similar in terms of structure. In terms of the sequence, it was so different. And we found that it is killing all the microbes. Now, where are you getting the sequences from? Those genome sequences which have never expressed, no RNA, no protein. This is the dark matter of genome. Likewise, we found novel antimalidor peptides. We experimentally determined that these computationally predicted antimalidor peptides from the yeast genome, when we tested them using the clinical strain of Plasmodium falciparum, we found that in presence of these peptides, 60% of the parasites are unable to enter RBCs. Likewise, we found a novel peptide that kills leishmania, and this was a very interesting story. It has just been, the patent has just been filed. So we said that tRNA never got a chance to express its own protein. It was just doing a kind of a social service for millions of years, but never got a chance. The molecules, but never got a chance to express itself as an mRNA because it is tRNA. It will not express itself. We know that. There are steric hindrances. But how about if we allow the tRNA to make its own peptide? Now, we cannot do this in a natural setting. We know this. So we took we bioinformatically, converted the tRNA sequence into an amino acid sequence equivalent using the genetic code table and chemically synthesized tRNA, encoded peptide, as we call it. And we found that at 40 or 30 nanomolar concentration, it is able to completely kill leishmania. And this is amazing effect that we never imagined from a tRNA encoded peptide. And the best part is that even if you increase the concentration of this peptide 10 times or even 20 times, it does not kill the human cells. It selectively kills only the leishmania. So now we are, we have filed it for patent. Likewise, we found some novel anti-breast cancer molecules and some Alzheimer molecules. We're working on that. And this has led to spinning of a company, Foresight Biotech, incubated at JNU. And so we do have some serious discussion with the funding managers to scale up the production of these molecules and a lot more. 2009, the first publication in the series was came up in Journal of Biological Engineering, and then I was invited to European Science Commission to deliver a talk on this work. And likewise, there are other organizations that were interested in our work, and so this has led to some global visibility, but a lot more needs to be done. So currently we have close to 500 molecular inventory for pharma and biotech industry. So the best part is that you own the IP of every molecule that you can make. You do not make it if it exists. So you have a lot of bioinformatics filters that prevent you from any such possibilities. So you make only those molecules for which there is no IP, and you make it and you can name it after your name. There are big challenges, of course, always. The whole idea was full of challenges when it was floated for the first time. But we have come out of that phase, and so we feel that we get a big message from a small atom that be positive, even if you're always surrounded with negative things. So thank you very much for your kind attention, and I'm happy to take questions, if any.
I'm highly thankful to Professor Dr. Pawan Dhar sir for sharing your valuable and insightful lecture on this complex topic, that is reductionism. And I strongly believe that this will definitely help our students as well as other participants to understand the correlation of the biotechnology with this computational biology. So this will definitely help them to further utilize your valuable and insightful lecture in their future research as well as in their career also. Thank you so much, sir, for this insightful lecture, sir. I'm heartily thankful to you, sir.
My pleasure, anytime, please. So if there are any questions, we can address them, or if we are running out of time, then maybe we can have it later.